All right, so where we ended up on Friday was here with vertebrate animals. If I can get this thing to start. Here we go. All right, fine. All right. This is a rather long PowerPoint. I've already uploaded it. One of the where you have actually converted it, but it's there. Okay? Download this PowerPoint from the place on the web where I've given you access to PowerPoint presentations, so that you can go back and look at it later if you need to. Where we stopped on Friday was talking about vertebrate animals. If you're working with vertebrate animals, okay, I think most of you know what a vertebrate animal is. I hope at this point. Studies you are not allowed to do include induced toxicity studies involving known toxic substance that would impair health or destroy life.
What if you're meant for like?
Is that what you're going to be doing?
Yeah, I mean we're putting.
That involves
in their water.
this, and you need to go back. You need to go back and read the rules very carefully and make sure that you're allowed to do that. Because if you go to the regional science fair with this, they're going to throw it out. Okay, if it's your project, so you have to choose a project, some spin-off that doesn't involve you doing this. Okay. Behavioral experiments with conditioning using adverse stimuli. Mother-infant separation. Induced or learned helplessness. Okay? Those are projects with vertebrate animals that are not allowed. Now you can do it with invertebrates, but you can't do it with vertebrates. They're higher level animals. They have a lot higher level of consciousness. Look, I'm not going to argue with you. I'm just telling you what's allowed. Okay? It's not my decision anyway. You know, I might agree with it or not agree with it. What I'm saying is you won't be allowed to take this project to a competition at many of these competitions. They won't allow it in there. Okay?
What if we're like not doing it for the competition?
Well, I'm hoping that you're going to be doing that. Okay? We're, as far as what we're doing is we're trying to follow these guidelines. Okay? They make sense. I mean, there are federal laws that undergird these ISEF rules. Okay? If you go outside that, then it's going to have to be evaluated. We'll have to convene an SRC and have somebody else make the decision. I can't make the decision. Okay? Studies of pain. So you're not allowed to inflict pain on vertebrate animals to study the results. Predatory vertebrate prey experiments. Now that doesn't mean you can't study it in the wild, but it means you can't sick a dog on a cat in a cage. All right? Just not allowed. What is the deal? Of course, around my house, the cat's in charge and the dog's afraid of the cat. But all right. Of course, my wife has this little bitty dog, you know. All right. We don't actually own a cat. We have a daughter that owns a cat. So, all right. Listen. No vertebrate animal deaths due to experimental procedures are permitted. Okay. You're not allowed to kill animals as part of your experimentation. That doesn't mean you don't sacrifice them to study animal parts. That's a different process entirely. Okay? All right. Studies designed or anticipated to cause vertebrate animal death are prohibited. Any deaths must be investigated by qualified individuals. So, say you're doing some research and you're not intending to harm an animal, but one dies. There has to be an investigation. If the death was a result of experimental procedure, the study will be terminated and will not qualify for any competition in the ISEF system. Okay? Behavioral observations of animals are exempt from SRC review if there's no interaction with animals. That's part of observing in the wild. You can do that. Okay? You can go into certain existing environments, not environments you create, but pre-existing environments and observe animals in the wild. That's allowed. No manipulation of the environment All federal or state game, fish, and wildlife regulations must be followed. Okay? Not only is that a ISCF rule, that's a state law. Okay? We just, we'd have a hard time getting around that. All right, research sites, homeschool, and in the field. So we talked the other day about a study in this pond, or what is it? Is it called pond? Okay. I don't forget the name of it. Where is it? Timms Creek. Okay. So there's an area over there that some of you may be actually working in. All right, that's fine. You can study what's going on there. You just can't manipulate what's happening there. Okay. An unnamed professor. He's not unnamed, but a professor I won't name. Some years ago, got in trouble with the Fish and Wildlife because he threw some sodium into the Pacolet River. How did they know? I don't know. They found out. But he threw a big enough chunk of sodium that it basically exploded in the river. Okay? But the problem is that not only does a concussion harm the fish, the addition of the sodium also makes the water become basic and more fish die. So anything like that is just not allowed. You can actually, under certain circumstances, shock the fish, but you can't drop a sodium bomb in there. All right. That's how they count fish. They put electrodes in the water and put a low-level charge through there, and the fish float to the top. They count them, and they swim away again, and they wake up in just a minute. It's kind of like getting hit with a taser, I guess. All right. Regulated research institutions. Some of you are working at Converse. Now, I ask you to check and see if they have an IRB. Has anybody had a chance to do that yet? No? Okay. You need to find out about that. If you're working in a research institution and they don't have an IRB approval for working with humans, if that's the case, you might be working with humans. It may be perfectly okay. But if they are working with humans and they don't have an IRB to approve that sort of thing, we have to convene one here. Okay? I forget what that acronym is for, but that has to do with the research institution itself. And that includes universities. Some of you are working in colleges and universities. I don't think we have anybody working with government research agencies. Okay, anybody in here would fit that category? Not government research agencies, all right. And private research laboratories. We do have some of that. Okay? But they have to have an IRB. Are you listening? They have to have an IRB. If they don't, we have to have it approved here. Or if it's not approved, you can't do it. Okay? Requirements for studies that take place at school, in your home, or in the field. Agricultural, behavioral, observational, or supplemental nutritional studies, and non-invasive and non-intrusive with no negative effect on animals' health or well-being, and require SRC pre-approval and pre-review and approval. Okay. If you're just studying water quality, that doesn't mean you have to have SRC pre-approval. If you're working with animals in the field, then you do. Does that make sense? Okay, so we'll have to set up an SRC. I'm just working on the IRB right now. I haven't gotten as far as setting up an SRC. SRC determines the level of supervision appropriate for the study, assigning a designated supervisor, veterinarian if necessary, qualified scientist if necessary. And if you're working with animal studies in the field, at home, or at school, a new form is required, 5A, vertebrate animal form. If that applies to anyone in here, now's the time to go to the website and download it. Okay? So the location for that, those of you who weren't here on Friday, if you haven't already looked at the video from Friday, this is the website you're looking for. This has all the forms you're going to need. Okay? Virtually all the forms you're going to need. There may be some rare cases where other things are required that fall outside this, but I don't think so. Okay? www.societyforscience.org/ISCF/document. Okay? Is anybody working with vertebrate animals? Okay? Then you're going to need that Form 5A. Please go there and download it to your flash drive right now. Okay? There's vertebrate animal form. It's right there, 5A and 5B. Okay? You're going to need that.
Why do we have to have all these different forms for that, like with demon and things? In case it gets
to prove that you're being ethical. Have you ever heard the story about Pavlov's dogs? Pavlov is a famous behavioral scientist. He was Russian. The research for which he is most famous was that there would be some kind of a signal, I forget if it was a buzzer or a bell, then a door would open up that would allow the dog to get the food. And he wanted to know, did they salivate at the sound of the bell? Could he train them to salivate at the sound of a bell? The way he figured that out was he cut off their lower jaws. Now, today, that's considered unethical. We don't do that sort of thing. Okay? That's the sort of thing. You are inexperienced as scientists. Okay? To make sure that you don't do something unethical that you might not even know you're doing is unethical. That's what, I don't expect you to know everything, but the forms help us to figure that out. That's what this process is all about, to make sure something unethical doesn't happen. Okay? All right, have you got this website and this? You got all this done? Okay. All right, so there's the vertebrate animal form. Requirements for studies at regulated research institutions. That's a lot of you. If you're working with animals, all right. Now, is anybody working at Commerce or some other facility where they're working with animals? No? If it doesn't apply to anybody, I'll move on to the next section. Does work with you? Okay. Must be approved by an institutional animal care and use committee. Okay? I don't have any control over that. They have to have an institutional animal care and use committee. Okay? It's not hard to set up. I can give you the federal guidelines for it. It's all in the federal statute. But they have to get all that. They're supposed to have that done anyway. They may have it. You may just not know it already. Okay? And a lot of times that's the case. The professor's already taking care of it because they know what they're doing. Okay? Or if the university has a different set of procedures, then it may be that because you need this, they have to take care of that. Okay? All right. The local SRC should review the project before experimentation. Since we don't have an SRC here, we can convene one if we need one. Okay? Your institution may have one already. Okay? The regional science fair, it takes place in February. Their SRC will review it anyway. It's a good idea to have it all covered in advance so that when they get it, you don't have any, you skate right through. Okay. All experimentation must follow ISEF guidelines. QS completes. That's the I forget what QS stands for. Oh, the qualified scientist completes the 5B. We just talked about that, and includes documentation of this panel's approval. Okay. And there's the vertebrate animal form 5B. So if you downloaded 5A a while ago, it automatically gives you 5B. Okay, I think they're part of the same thing. At least that's what I think it is. I don't use them very much myself because I don't deal with animals too much. Okay? All right. This is one that almost all of you will have to have. Okay? Not biological agents, but you'll have to have a hazardous form. Okay, you turn in. This is biological agents. So if you're working with any kind of biological agents, bacteria, okay, well, here it is. Microorganisms, including bacteria, viruses, and fungi. Recombinant DNA, human or animal fresh frozen tissues. Somebody's working with, somebody has to sacrifice, who has to sacrifice an animal to study what they want to study? Is it you? Okay, so this applies to you. Okay? Studies must have prior approval. So your institution may already have all that. Okay? Most studies are prohibited in a home environment. That's not you. Studies intended to genetically engineer bacteria with multiple antibiotic resistance are prohibited. That would be prohibited in most research institutions. Lab studies utilizing MRSA, VRE, KPC must be conducted in a regulated research institution under documented IBC approval and review. Okay, I don't think that applies to what you're doing though, right? Megan? That doesn't apply to you, right? Okay. Let's see. Required of all PHBA projects defines potential level of harm, injury or disease to plants, animals, or humans. Involves assignment of biological agent to a risk group, determination of level of biological containment, assessment of expertise of adults. That's really critical right there because you don't want somebody teaching you who don't know what they're doing. Assignment of final biosafety level. So that means, what do you have to do to make sure that you're safe working around these things? Okay, are you just going to have to, wear gloves, or is it something more than that? You have to have a respirator. Do you have to wear full? You can't. I don't think students are allowed to work with full gear anyway. But all right, BSL 1 level studies can usually be conducted in a high school or college teaching laboratory. BSL 2 level studies are usually conducted in a regulated research institution. And these higher levels are prohibited in ISCF projects. So students aren't really supposed to be working around this stuff anyway. Does that make sense? All right. Form 6A, potentially hazardous biological agents form, required for most projects involving microorganisms and for all projects involving rDNA and fresh human and vertebrate animal studies. Potential hazardous biological agents risk assessment form 6A. If you're working with that, download that now if you haven't already. And if you need to go through and look at the complete rules that govern these things, that web page I gave you a minute ago has a complete set of rules. Okay? And for this sort of thing, Dr. Terry's probably better equipped to answer some of these questions for you than I am. Okay? I don't work with recombinant DNA. All right? I've done it, but I don't work with it typically. Okay? Exemptions from prior SRC review and no additional BHPA forms required. So these are the things that you don't have to have prior approval for. Baker's and brewer's yeast, except the rDNA studies. Studies using Lactobacillus B. Lactobacillus B. thuringiensis. Dr. Terry, help me if I mispronounce these things. Nitrogen-fixing bacteria, oil-eating bacteria. Okay, good. Algae-eating bacteria in natural environments. So if you're working with them in the regular environment outside in a field study, that's okay. Not exempt if cultured in a petri dish environment. So if you work with them in the lab, they're not exempt. If they're in the field, they are. Does that make sense? Studies of mold growth on food items if experiment terminated at first sign of mold. Okay, so you can study, say, what makes a good culture for mold, but you can't continue to grow it through its lifespan. Okay, which is interesting because my oldest son violated this rule and I didn't know about it. That was way a long time ago because he studied mold in an elementary school project. Okay, studies that are exempt from studies that are exempt from SRC review, scientific review committee. That's what SRC stands for. You're required to have a Form 3. This is to show that they're exempt. Okay, you have to basically justify your exemption. Studies involving protists, archaea, and similar microorganisms. Did I pronounce that right? Archaea bacteria. Okay. Research using manure for composting, fuel production, or other non-culturing experiments. Studies using commercially available color change coliform. Is that right? Coliform? Is it coliform or chloroform? Coliform? Water test kits. Okay. Studies involving decomposition of vertebrate organisms. Forensic studies. So, yeah. Okay. Studies involving unknown microorganisms. All right. This is remember you got the 4 levels. BSL 1 if organisms cultured in a plastic petri dish. Or culture dish remains sealed throughout and the culture dish remains sealed throughout the experiment. Culture dish is disposed of in an appropriate manner, which means it's basically killed off without any chance for human interaction. Okay. Let's see, BSL 2. All right, that's what constitutes BSL 1. I'm sorry. BSL 2 then is that hazard level that if you're working with these kind of things and the petri dish is open. So if there's you probably have to have some kind of respiration apparatus or something, or some way to isolate the culture, keep it from you from being exposed to it in that case. Experiments with BSL-1 organisms can be done in a BSL-1 lab with a qualified scientist or trained, what is this DS? I forget. We talked about it on Friday. This is like a college graduate student who's working under the professor to help with the lab experiments. Okay, that's the kind of category that falls into. Experiments with BSL-2 organisms must be done in a regulated research institution, so you can't do this in a high school lab. And it has to be with a qualified scientist. Tissues. If animal is euthanized solely for a student project, the vertebrate animal study, I think this means it requires its approval. Okay? So I think that's what Megan's working on, right? Isn't that you? Killed. Okay. So they have to have this committee approval here. Okay. If the animal is euthanized for a purpose other than a student project for tissue study, and this is not really very clear, but I think what that means. Well, maybe I better be sure and go back and look at the actual, or maybe you and Dr. Terry should go back and look at the full research document for ISCF, the full thing, the big thing, okay, not just the student handout. Okay, this is telling us more about what a BSL level study 1 or 2 is based on. Let's see. All studies with human or wild animal blood are BSL-2. Studies with domestic animal blood, BSL-1. Studies with human body fluids, which can be associated with a person, must have IRB approval. I'm not sure how that would stand up. You'd have to have some really careful evaluation of that. Exempt is BHPA tissues. Plant tissues are exempt. Plant and non-primate established cell and tissue cultures. So these are things that have already been worked with, not something that's taken out of a sacrificed animal that's not already an established cell or tissue culture. Fresh or frozen meat byproducts, pasteurized milk, eggs, or other things from a grocery store, restaurants, or packing houses. Those are exempt. Hair, sterilized teeth, sterilized. Why would it have to be sterilized? Is anybody listening? You with me? You don't know why? Yeah, you don't want to get the tissue, the blood and stuff, the human, what? Yeah, all those pathogens in your mouth. Yeah. Okay. Fossilized tissue, because fossilized tissue. That's interesting the way they use that. The tissue doesn't exist anymore if it's fossilized. Or archaeological specimens. That's interesting the way they use that. Prepared fixed tissue slides. Okay, so that's obviously it's fixed means you basically aren't coming in contact with it anyway. Form 6B is required for all projects using fresh or frozen tissue, primary cell cultures, human and other primate established cell lines and tissue cultures, blood and blood products, and body fluids. Okay? There's the 6B. Hazardous chemicals, activities, or devices. Okay. If you're doing pure research, you're going to have one of these forms regardless. Okay? My experience with this is better to be safe than sorry. Go ahead and do it. And this involves anything working with chemicals. And that can be something as simple as the stuff you'd use to wipe off the lab tables. Okay, in a biology lab. Okay? Hazardous equipment. Well, does that involve glassware? Glassware can be hazardous, right? What about something that's electrical? Just about anything you can be working with in the lab, you're going to need this form filled out. Okay? DEA controlled substances. Okay, so drugs. Prescription drugs, alcohol and tobacco, firearms and explosives, any kind of radiation. General rules. Studies do not require prior SRC review and approval. All studies require risk assessment documentation on Form 3, and that's the one for hazardous materials and equipment. DEA controlled substances require a qualified scientist, so you couldn't do that in the high school. All of the studies require a designated supervisor. Okay? If you're not working under a worksite mentor as your research mentor, then Dr. Terry or I become your designated supervisor. Okay? We may also be your adult supervisor. Nobody's working with drugs, are they? I haven't seen anybody's projects where they're working with drugs. Maybe. Are they? You know if they're on the list of controlled substances, DEA's list? Most likely. Okay. Then you're working under a qualified scientist, so you're okay there. Anything on Schedule 1 requires a DEA protocol review. Okay? You make sure you're aware of what Schedule 1 is and just check that. Okay? I don't think anybody's working with firearms or explosives this time around, are they? No? I've had students do it. Okay? So we had SRC approval at the Regional Science Fair, and they actually went to the International Science Fair. So now, obviously, you can't purchase alcohol and tobacco. Okay? So that's something that has to be purchased and controlled for lab use by an adult supervisor. Okay? You're going to need to, in your research paper, explain how it was acquired, how it was used, and how it was disposed of. Are you clear on that? Okay, that should be in your materials and procedures section. How it was acquired, how it was used, and how it was disposed of. It is not allowed. Now, this is my rule. Okay? If you're working with one of these substances, I don't think anybody in here is, but if you are, you don't get, I won't even give you a grade on your paper if it isn't in there. Got it? Almost any chemical you work with, you need to include in there how you're going to dispose of it properly. Okay, so you need to do a little research. If you're working with an acid, what's the right way to dispose of it? If you're working with a base, if you're working with some DEA-controlled substance, what's the right way to dispose of it? Okay? Some things you can dispose of in the waste stream, meaning down the sink, basically, and some things you can't. But you must follow EPA guidelines on that. Okay? EPA guidelines become laws for you. Okay? All right. It is allowed, ethically and legally, to produce ethyl alcohol, as long as you're following alcohol, tobacco, and firearms regulations. Okay? So that is actually an allowed project. You wanted to try and find the best way to produce ethyl alcohol for use in a car, that'd be allowed. Okay? But it must be strictly monitored. Make sense? You got to have somebody looking over your shoulder all the time. All right. Now, if you're doing more than just a small sample, we'll have to get alcohol, tobacco, and firearms to approve it. Got it? If you're actually trying to produce it and have to run a car, we'd have to have a special permission to do that. Prescription drugs. Nobody's working with prescription drugs in here. Hazardous chemicals. Almost everybody is. If you're working with hazardous chemicals, make sure you have a copy of the MSDS for your chemicals. Okay? Include that in an appendix. Got that? Yep. Yep. The federal government, I hope everybody's listening, the federal government requires that you know or be allowed to know the hazards of the chemicals you're working, hazards of the chemicals you're working with. Okay? And from my perspective, you must have a copy of an MSDS in your documentation. I would suggest you make it an appendix, okay, to your report. Got that? Now you can, like for instance, they come out, might print an 8.5 by 11 sheet, might be 6 pages. You can do 2 pages per sheet or something if you want to copy it out that way. Just as long as it's in there and it's readable. Does that make sense? All right. And that's a really good thing. We talked about what goes in the appendices of your project report. That's one good thing to make sure you put in your appendices, okay? Let's see. I guess this is a pretty general thing here, but I guess if you're using a hammer, you don't have to include that as on your list of hazardous devices. Okay? Even though you can obviously smash your thumb. All right? But anything beyond that, you've got to have this listed on this form for hazardous materials and devices. Let's see. Different forms of radiation have to be included. I don't think anybody's working with radiation here, are you? Unless you're working in a bio lab where they're doing cleanup with radiation. Nobody in here? I don't think so. There's different types of radiation, okay? Most of the ionizing radiation would include things like X-rays or gamma rays. That's non-ionizing. Ionizing would be things like alpha particles and beta particles. Form 3 was required for all projects involving these DEA controlled substances. That involves things, that's all the chemicals, okay? Prescription drugs, alcohol, tobacco, hazardous chemicals, hazardous devices, hazardous activities. If you're climbing on a ladder, that's a hazardous activity. Okay? Better to be safe than sorry, right? Or radiation. And there's risk assessment form 3. If you're doing a pure science project, you must have this. If you're doing an applied science project, you may also have to have this. Okay? If you're working with horses, you got to have this. If you're working with bacteria, you got to have this. Okay? If you're doing water testing, you got to have this. Got it? Climbing on a ladder, got to have this. If you're using a hammer, not necessarily, unless you're breaking glass. Then you got to have it. All right. I know that was the boring part. The fun part was at the beginning, right? Okay. Any questions about the forms you got to have? Has everybody downloaded the forms that they need? Okay? All right. Make sure that if you are working in a college or university or at a business sort of facility or something like that, professional facility, you go through this stuff with your project mentor sometime this week. Make sure you've got it covered. All right? Do not take this set of papers, put it in the hands of your research project mentor and say, here, do this. That is not their job. Okay? That's your job. Your job is to find out, and they can help you with it. I'm sure most of them will be willing to do that if they have the time. They can answer questions for you. It's your job to get the paperwork filled out for their signature. Does that make sense? You are the secretary. Got it? Any questions? Huh? Everybody's clear on that. You're not going to go and ask, just, here, do this. That's not their job. Your job is to make sure you do as much of the legwork as you possibly can for them. And then ask for their signature. Got it? Any questions? All right. Not hearing any, we will call this meeting to an end. No, we won't. We'll just stop this video.